Today we are going to be studying mitosis using the compound microscope in the onion root tip. Our objective is to identify the different stages of mitosis. Different stains are used when staining the chromosomes to see their division. We will be using Schiff's reagent. Schiff's reagent relies on a reaction called the Foilgen reaction. The Foilgen reaction uses hydrochloric acid. The hydrochloric acid hydrolyzes the pentose in the DNA. These aldehydes then stain with the Schiff's reagent. Onion root tips have been grown for the purpose of our experiment. Once they got to about 2 centimeters long, we cut them off and put them in aceto alcohol, which is a fixative, for between 24 to 48 hours. Onion root tips are examples of meristematic tissue. Meristematic tissue is actively growing tissue, which means we're going to find all the different stages of mitosis. The tips have been cut off of the onion and put in the fixative, as I explained earlier, for 24 to 48 hours. In the methods now, the root tips are exposed to hydrochloric acid. This is for the hydrolysis step. So there's an example of our hydrochloric acid, and that is for 8 minutes. The root tips are then stained in the Schiff's reagent for 2 to 4 hours. This gives them a very pink-purple color, as you can see in this test tube. The root tips have been stained in the Schiff's reagent and have since been rinsed in water. I'm now going to show you how to put them on your slide. We take a drop of acetic acid, drop it on the slide, then we try to catch a root tip. Then we get a cover glass, and you make sure that you get no air bubbles by using the tip of the needle to let the cover glass fall slowly onto the root tip. Once it's on the root tip, you can use the back of your needle to tap it. You can also use your pencil. This separates the cells so that we can see the different stages of mitosis. And then we use some filter paper to just blot off the excess acetic acid that has slipped out on either side. And your slide is now ready to view under the compound microscope. Okay, so I have my slide that I've prepared on my microscope stage. I started at the 10 times to find my cells. Once I found the cells, I moved on to the 40 times objective, which is what you now see on the monitor. Okay, so if we look at this part of the slide, it's the same slide that we were dealing with. We can see here we have a prophase. Here we have a metaphase. And then if we just go down a little, we see an anaphase and a telophase. So with that being said, we have covered all the stages of mitosis, and this is a very successful slide. And that concludes the practical on the 4 stages of mitosis.